Hello and welcome to the Chapter 20 video from the AP Bio Exam Review video series, Phylogeny. Okay, when we start with phylogeny, we're going to start by talking about taxonomic categories and how they indicate relatedness. So Linnaeus was the first one to actually start classifying animals or organisms, and he classified everything into plants and animals. Excuse me. And so since then, we've gotten a little bit more precise with our language. In fact, a lot more precise with our language. But we're still going to use a lot of the same terminology that he originally came up with. And so we have kingdom, phylum, class, order, family, genus, and species. And you need to remember these in the proper order. And I don't care what acronym you use. I use Ken poured coffee on Fran's good shirt as to indicate K, P, C, O, F, G, S, right? You're welcome to come up with whatever acronym you want, but you need to remember the order that they go into. What does it actually mean? Well, domains, I mean, kingdoms are one into domains, but these guys down here are very, very large groups of very loosely related organisms. And so when we talk about kingdom Animalia, that's everything from mosquitoes to blue whales to earthworms to kangaroos to alligators. Everything that is an animal goes in here. Tapeworms. And then as we move up, the groups are much, much smaller, and the organisms that are in them are much more closely related. So Phylogeny, these are all your cats. Okay, that's a pretty small group of organisms. These are just your panthers. This is just that one species of panther, and remember that that just means that they can all reproduce to produce fertile offspring. And so the further we are down here, it's a bigger group, loosely related organisms. You need to know how systematics is used to develop phylogenetic trees. So to take that diagram, the kingdom, phylum, class, order, family, genus, species, we're going to just look at a subset of it. And here we have order, family, genus, and species, and it's just saying that within this order we have multiple families. Okay, and in fact there's way more than just the ones we have here. And then within this family we would have different genera. Okay, it's plural of genus. And then within that genus, we would expect there to be multiple species. And so when we look at our phylogenetic trees, we can, we're getting to the point now where we can associate the taxonomic relationship to the phylogenetic tree. And that's something that back when Linnaeus started obviously wasn't even an option. He just looked at things and tried to put them together. But now that we're using so much DNA evidence, we're able to assemble this in a pretty straightforward way and connect the trees with their taxonomy. All right, you need to know how to construct a phylogenetic tree that represents the processes of biological evolution. So this is why I made you do that packet where you had to put together, find all those characteristics and build those trees. So when we look at legless lizards versus snakes, when we talked about that at the very beginning of this chapter back in lecture, and I said, look, it's a legless lizard, and some of you were like, well, what's the difference between that and a snake? Isn't a snake just a legless lizard? And we talked about no. Lizards actually legless lizards developed or lost their limbs completely separately from the loss of limbs with snakes. And so even though they look very similar, they actually had that evolution event separate. And so here is convergent evolution, where we are able to have the same structure, but not necessarily from the same evolutionary event. But looking at this, we're able to see, okay, this is where we would have lost limbs. We put these little tick marks on our phylogenetic trees. Your textbook does mostly the trees that are, rectangular like this. College Board may ask you to draw a phylogenetic tree, in which case draw the triangular ones that look like this. They're not really triangles, but I see triangles in there. I think that they're an awful lot easier, and then you can just mark here or here where you think characteristics are gained or lost. They may give you like a blank one, a blank structure, where you would get this part, and then you fill in these parts over here. which is fine too. You can handle that. It's just a matter of sometimes figuring out how to draw these things is really complicated, and so that's why I suggest you just do the triangle ones. So how do you actually do this? Well, just like I had you guys do in class, you have your organisms and you have your characteristics and you make a chart and you say, there's different ways to do this. In this chart, it's zeros if they don't have the characteristic and 1 if they do. It might be check marks or X's or something like that, but regardless, you have some way to delineate who has characteristics and who doesn't, and then you just put them in order and you say, okay, well, only one thing has hair, so that's going to be the least related, and that's why we're down here. If 2 things have the amnion, all of these guys, or here the turtle's got all of them, the leopard has all of these, you just kind of line them up so that, right, the turtle is above the leopard, the frog is above, the bass is above, and so forth. Now, College Board isn't always very nice, so they won't have them in quite the right order, so you have to look at them and just decide, okay, the first thing to branch off here is going to be my lamprey, and then after that's going to be my bass, and then so forth. But you go through this process just like we did in lab of making these tick marks, researching these features. Now, they aren't going to ask you to know these features. That's not a part of your curriculum. In class, I made you guys look them up. But they should give you a chart that indicates what organism has which features, or at least give you text to read to indicate to you which organisms has these features. You need to know the difference between those 3 domains, their similarities and their differences, or those 3 domains of life. All right, so, I talked in class about how domains didn't really exist when I was in high school. They kind of did, but they hadn't made their way to the textbooks at that point, at least not the high school ones. And so right now we're dealing with these 3 domains. And then within those domains, we still really only deal with kingdoms for plants, fungus, and animals. That's why they're in blue there. And then with domain archaea and bacteria down here, these are just, don't memorize this, you just need to understand that here we have domain archaea. These guys are kind of I'm going to call them our weird prokaryotes. They're the ones that live in pretty extreme environments. Domain bacteria down here, we know what bacteria are. These are the ones that we find all over the place, that are on our skin, that some of them make us sick, gram positive, gram negative, all those kinds of things. Eukarya up here, those are the ones that, of course, have those membrane-bound organelles, have the true nucleus. Many of them are unicellular, and so we used to put those into the protist category. So like all of these as well as this used to be considered protists. We still can call them protists, but they're not their own kingdom anymore. And then algae used to be lumped in with the plants, and then it had its own group, and then now the red and the green are even separated out as well. But we're just going to remember that despite all of this branching, that we are able to identify some features that are true for all organisms. And that's when we talk about continuity of life. And so things that are, things like we all work off the same genetic code, and that same that code, the A's and T's and C's and G's, the translation of that code into protein works off the same language. We talked about the codon chart. An AUG is always methionine, it's always your start codon. Whether or not you are a human or you are a fungus, that is the same. And so that continuity of life is kind of implied in this kind of a chart, is that how are these organisms related? Well, we can trace them all back to what they claim is a common ancestor of all life that has these features of the same genetic code, translation is the same, and that's indicating to us that all organisms branched from this common ancestor. So with regard to what I was just saying about proteins being, the translation of proteins being conserved, we can look at other proteins, not just on translation, but proteins such as the proteins for protein synthesis, glycolysis, Krebs cycle. These guys tend to evolve very, very, very slowly. And that makes these genes very useful for understanding those deep branches in the history of life. Ribosomal RNA is another great example of something that changes very, very, very slowly. And so when scientists are trying to build these trees, these phylogenetic trees, these are the kinds of proteins that they look at when they're trying to assemble the data. So what we did in class is we looked at hemoglobin, because that's a good one for mammals, because that particular gene changes so very, very slowly over time that we're able to draw a lot of conclusions about relationship. Now, the flip side to this is that whether you look at protein synthesis proteins, glycolysis proteins, Krebs cycle proteins, ribosomal RNA, the flip side is when you look at these different proteins or protein groups, you often get different trees. And so, despite the fact that we have these very slowly changing genes, they don't all point in the same direction. And so that's something that is a bit of a confusion in this branch of science. So that's phylogeny. That's how we're building trees, how to read trees. I hope you found that useful, and I will see you in class.